So, courtship behaviour is vital for species to recognise members of their own species. So, there's no point doing a display or dance or a call that attracts another species or also can attract potential predators. So, it's definitely vital to recognise your own species. It's one of these sort of things that's often forgotten in this, that the displays, the calls, whatever it is highly specific to that species. The displays may look virtually similar to us, but to the individual organisms, they are highly specific and immediately recognizable. Another reason courtship displays are used is to identify mate of a breeding age. You do not want to attract immature or non-sexually viable mate. So there's no point displaying and attracting a younger member of the species. There's also the forming of a pair bond, which is very important for many species. A pair bond will ensure or help to promote species and offspring survival. The pair will take responsibility, equal responsibility usually, for looking after the young, and that means it's increasing its survival chances. Many species do this. A couple of examples are beavers, wolves, otters. Swans do as well. The other sort of form is where it's just herd mentality, and there will be mating within there, but usually it's the strongest that has a harem of females that it will protect, such as deer and things like that. But they will not form a pair bond. Mating will be done seasonally. Also, another reason for courtship is to synchronize mating. This will ensure that offspring are born in relatively close proximity to each other at the same time. So the survival chances of the individual offspring will be increased by the majority of the organisms giving birth. The lion, for example, in the wildebeest herd, has so much choice that the individual is more likely to survive because there is now 1,000 young to eat as opposed to 3 or 4. And the main reason that courtship behavior is used is the imperative of all organisms to pass on DNA. Using synchronized mating will maximize the offspring's survival chances, as I've just mentioned, and also they will ensure that food is always abundant. There's no point giving birth in the middle of winter because there's less food. So the synchronized mating and courtship displays will all be done at the correct time to therefore mean that the offspring are born in late spring when food is becoming abundant. Also, they've got the entire summer to store food, and it's a warmer weather. There's less chance of infectious disease, that sort of thing. So, how is courtship done? Well, it's actually really simple. The male carries out an action. 90% of the time, it's the male. Very rare for the female to initiate a courtship display. It's usually the male. It'll be a display of colours, patterns, maybe a call, maybe an action, something like that. But whatever it is, we're going to call it the action. So it could be, like I said, a display, a call, but it's an action. This is detected by the female as the stimulus for this female to respond. She will do something in return to acknowledge the male has done that action. This is the stimulus for the male to carry out the next actions. This could be something like the male dips his head and the female responds by dipping hers. He then shakes his head, she shakes hers. He then ruffles his feathers or fans his wings, and she responds in like manner. It could be he gives a gift, she accepts the gift, and then he maybe preens her neck and she responds by doing the same. But the thing is, there is a stimulus, and this is repeated. It could be up to 30 different actions that are done. And if it's all successful, the net result is mating. But if it's not, or it gets disrupted, the male will have to start again, which is why courtship displays often fail, because the female is not impressed, or it's disrupted vent, or he makes a mistake, because it can happen. And the whole action and response here is called the stimulus response chain. So male carries out action, that's the stimulus to the female to respond, the male carries out the next action, etc., etc. So that passing on of genetic material means that we have genetic diversity. And it's quite simple. It is the greater the number of alleles, the greater the genetic diversity, therefore the better adapted to the environment there is the chance of. So it's all sort of interlinked to the mixing of the genetic material is vital to bring out diversity, which in turn is very good to improve chances of survival. We have been modifying genetic diversity for thousands of years. We've done it through selective breeding, where you keep only the ones you wish and you cull or kill the ones that are not viable. So in the fish example here, the non-viable fish are all killed off or not allowed to reproduce. You don't have to kill them. but they are culled from the breeding population, and only the ones that we deem to be beneficial or to look nice or to taste nice or whatever, we're allowed to breed. And that is then weeding out what we see as undesirable genes. But unfortunately, the effect of this is it reduces the genetic pool, so we can actually end up with genetic abnormalities coming in, such as the breeding disorders in pugs or deafness in Dalmatian dogs. This can also happen naturally in the environment. It's not just our control that does this. This could be caused by something like the founder effect, where a few individuals of a species colonize a new area. So birds get blown off course by a hurricane and end up in an island where there are very few alleles, so inbreeding can then start to occur. And when that happens, we get less and less genetic diversity, therefore less adaptation, and the species has difficulty adapting to the environment if it changes. If it's stable, it's fine, but if it changes, there is a problem there. Another thing that can limit genetic diversity is genetic bottlenecking. That's where a large population is massively reduced in a short space of time, maybe due to natural disaster, maybe due to overhunting. So what happens is we lose genetic diversity. Population suffers a big hit, numbers drop drastically, and that leaves a lot fewer individuals, reducing the amount of genetic material available because we've just got rid of most of it. And we end up with a reduced amount of alleles, which means inbreeding is a lot more likely to occur. And that's just going to compound any genetic problems within that limited population. There's an example of genetic bottlenecking with the northern elephant seal, which was hunted virtually to extinction. There was only 20 individuals left by the end of the 19th century. And since then, it has been protected and has recovered. But the problem is there is a massive amount of genetic problems within this because they are so reduced in number. Only 20 breeding individuals means that they've very much compounded the genetic problems. So we've now got around about 30,000, but the bottlenecking has occurred because there's less genetic variation. Whereas the southern elephant seal was not so intensely hunted and it's got a lot more variation in its allele frequency. And to finish off on, we're going to look at DNA hybridisation. This is how we can use genetic material to determine how closely related a species is. It looks very complex and it is quite an intense sort of technique, but it works really well. It all requires, though, two separate species. We want to know how closely related they are. So what you have to do is you have to split the DNA up and allow it to make the hybrid. And when it makes the hybrid, you can see how well linked the bonds are. The more hydrogen bonds that form, the more closely related the species is. If it makes only a few hydrogen bonds, they're distantly related. So we've got to break the DNA apart by heating it, allow it to bond back together and make sure the DNA hybridizes. So we ignore any that stick back to the original DNA. We only want the green and the blue here to bond together, for example. And then we can check the amount of hybridization and the bonding by slowly heating it and seeing how long it takes to fall apart. So first off, to make the hybrid DNA, you need to isolate the genetic material. So you need to cut it up, purify it, and then you radioactively or fluorescently label one section of DNA, so one species of DNA. What you then do is heat the DNA until they separate. And you do this together. So both sections of DNA, the radioactively labeled double strand and the non-radioactively labeled double strand, are heated in a big heater together until they separate. And as they cool, they will bind back together. Now, there's got three options here how they can bind. Either the original DNA can form with the original DNA in one species, and the original DNA can form with the original DNA in the other species, or one strand from one species and one strand from another can form together. And that's the one we're looking for. So we allow them to cool down, the DNA to cool down, and we're looking for that 50% labeled because one strand will be radioactive, the other strand will be not. So if it's 100% radioactive, we don't want it. If it's 0% radioactive, we don't want it. If it's 50% radioactive, that's the one we want because it's got one strand from one species, one strand from the other. And that is DNA hybridization. We've now made a hybrid section of DNA. Now what you do is you take that hybrid DNA and you put it into another heater and you ever so slowly increase the temperature. you slowly raise it in stages, maybe going up a degree at a time. And what will happen is the hydrogen bonds will start to break because the temperature will start to disrupt the hydrogen bonding. They're quite weak and they're temperature sensitive. So the temperature slowly increases. You check it every degree, every half degree, and slowly what will happen is the strands will start to separate. Similar species may have 90% of the hydrogen bonds there formed because of the base pair sequencing, A to T, C to G. So there's 3 hydrogen bonds on the G and 2 on the T. So if there's an A and a G opposite each other, they won't form a bond. So they're less likely to be a similar species. So the more hydrogen bonds, the more closely related the species is. The higher the temperature it gets to before the DNA separates, the more closer they are. If it gets to 90 odd degrees and they still haven't separated, they're probably the same species. If they separate at 30 degrees, there was only a very few hydrogen bonds there, so they were not very closely related. So the greater the bonds, the stronger it is, the better it is at withstanding that high temperatures. And just to reiterate that, the higher the temperature before they separate, the closer related they are. If we did this with humans and chimpanzee DNA, it would survive probably up to about 95 degrees before it finally separates. Whereas if we did it with human and slug, it would probably separate at about 20 to 25 degrees. I'm not exactly sure when it would be, but you can see the difference there. Hope that helps. Thanks a lot. Bye.